after the event. You're all muted by default, so if you have a question, then you should just feel free to type it in the box, and I'll try my best to get round to answering specific queries as and when there is time. So I'll be talking today about in silico methods in fragment-based design, and as a case study I'll be using some data from Protein Kinase B. Just move on. So this is just the general webinar guide. Like I said, you can type your questions in the question box of recording. So if there's anything specific you would like to find out, again, just type that in the question box. I'll spend about 5 minutes talking more generally about fragment-based design and Protein Kinase B, then a little bit about fragment collections, about how you find and filter those, and then on to the real meat of the webinar, which is docking and scoring fragments, of course using BioSolveIT's products. And then a little bit about, if you do have fragment hits, how you can go about using in silico solutions to find better or more selective fragments. So first, a little bit about me. My name's Alistair Donald. I'm an organic and medicinal chemist by training. I started my career with Cancer Research UK back in 2002, which is where I worked on Protein Kinase B. I then worked for Chroma Therapeutics, spent some time with Argenta Discovery, and I've been with BioSolveIT for almost 2 years now. So as I mentioned, while I was at the Institute of Cancer Research, which is where the drug discovery part of Cancer Research UK is located, we had a project that was running in collaboration with Aztecs Therapeutics, who are of course pioneers in the fragment-based design field, and we were working on not only fragment-based approaches to inhibitors of protein kinase B, but also some more traditional structure-based approaches and ligand-based approaches. And this was a very successful project and resulted in a licensing deal with AstraZeneca. And there's a compound currently in phase 2 testing based on the work which I undertook at the Institute. There's another good reason for picking PKB. This is a slide showing some data from GSK where they looked at hit rates across kinases, and you can see in the top corner here AKT1 and 2 are just pseudonyms for PKB, and they've got relatively low hit rates against AKT in these fragment screens. So one of the major challenges with not only Protein Kinase B, but perhaps kinase programs in general, is selectivity against other kinases, and this was definitely an issue we faced, especially with Protein Kinase A, ROC2 and GSK3 beta. And I'll talk a little bit about how we overcame those issues, and more specifically the strategies that we employed to do so. And alongside that, I'll focus more on what computational techniques we could have used. We didn't use these techniques, at least at the Institute. Aztecs did the computational part of the project. What techniques we could have used to help ID good fragments. So this is a general schematic of ATP binding. In this case, it's an ATP analog which binds to the hinge region. And one way we might think about, focusing our screening collection is, of course, just to fill it with the typical kinase hinge binding structures. And I'll show you how you can do that using one of our tools. Of course, if you don't know, the kinase hinge just connects the C-terminal and the N-terminal lobes of the kinase. And I'll be using as the structure in this case 1O6L, which is an active form of protein kinase B in complex with a peptide substrate and with this ATP analog. So, first off, enriching your fragment screening collection with potent and selective fragments. So first, before doing that, I'll maybe launch a little poll to ask you about the current status of what you're doing in your work, very generally, and that it's just useful feedback for me. If you could take a moment to click a selection, and we'll try and focus in on making it most appropriate for this audience. So we seem to have quite a few currently working in fragment-based design. Hopefully we can keep you entertained with BioSolveIT solutions. So I'll just close that poll and going to move on with showing you how you can use our tool Mona to do some smarts filtering and molecular property filtering. So because of course if you have a limited screening capacity, what you want to do is fill the top of your screening funnel with as many good quality fragments as you can. And not just good quality fragments, also if you can bias your selection towards soluble fragments or selective fragments, or improve the odds of, at the bottom of your funnel, having 10 or so good quality hits. So one way of doing that is to take what's commercially available and to filter it. And what I'll show now is just how you can really quickly use the tool Mona to customize the selection for your specific target, whether that is by combination of physical chemical properties and descriptors, or on the basis of some kind of pattern of chemical connectivity that you might expect to hit a kinase structure. So what we're going to do is just quickly download a couple of catalogs just to keep things moving quickly, and then filter those by the Aztec's rule of 3 type criteria. And this, I'll show you how you can add in extra filters should you so wish. So I'm going to use just a simple collection of smarts patterns. First one describing amino pyridine or pyrimidine type structure. The second one pyrazole or indazole type. And the third is just a simple aromatic carboxamide. So I'll just open up the Mona tool and show you how easy this is. Mona is a free tool for academics, and recently was updated and is really quite powerful. So I just click on File and Add Molecules. In my webinar folder I've pre-prepared a couple of vendor catalogues from Enamine and Maybridge. Just click on Open, and then Mona asks me, okay, you want to import these molecules, would you like to add some molecular properties while you're doing so? And here I've just added some very standard molecular descriptors. This then of course means that when I want to filter I don't have to go back in and calculate those, I've already done so. If you want to add an extra one, I just click on the drop down and say, okay, I want to learn about a number of aromatic rings, and just click on OK. And the Mona will then import those for you, and while doing so we'll eliminate any duplicates. So if I select my combined two catalogues and then go to the Molecule Sources tab, you can see that the Maybridge SDF that I've imported has listed in the duplicates column that it's got 2 duplicates. So what I'll now do is just go to the Filters tab. This is where we can achieve filtering, and I've pre-prepared a couple of filters. So I can go to Import Filters, and again, these sit in my webinar folder. I've based one on a pattern of the kinase smarts I just showed in the rule of 3, and I'll click on Open, and that then adds these to the right hand side panel filters. If I select the first one as the kinase smarts, I just select it, click on the little left pointing arrow, that just adds my smarts filters, and I can choose to include or exclude on the basis of these properties. So if I wanted for some reason to exclude everything, I would just click on the button. That would exclude the first one, but I want to actually include these three smarts patterns. But I want to make sure that these are independent of each other. I would just want to match at least one of these filters, and so I have to go to the little checkbox here and modify the number of filters that I'm altering. I don't know if that's very clear for you, so I'll just use a spotlight, and this button here I want to change to 1. So then, having done so, I just click on create a filtered set, and Mona will quickly go through those and strip out everything which doesn't match those smarts patterns. So I can go and take a look at those, and you can already see we've got lots of things that look much more kinase inhibitor-like. So just to do the second filter, my rule of 3 filter, I can then do the same thing, just select the filters, add those, and yes, I want to replace. And of course, you kind of combine these filters into one. I just did them separately for clarity, and I've managed somehow here to duplicate acceptor, so I'll just delete that one. Again, I can include or exclude, but here I want to match all 4 filters, just to exclude everything that doesn't match all the rule of 3. So all filters are being matched. It's already telling me that I'm going to end up with 197, so that's quite nice. And I click on create a filtered set. I've got 197. So I can now select that subset and see them within Mona, and I'll just export those. And to do that, I just right click on that set, choose export set, and then I can save my file ready for my docking experiments. Put it in the fragments folder, kinase set. And that's really it's really that easy to do. Of course, if you want to filter by smarts patterns, generating smarts patterns can be a challenge. We also have another tool available on our website called Smarts Editor, which is a really nice way to interactively draw, edit, and evaluate smarts patterns. It's also free for academics. So super. So now we've got our miniature set of molecules for our docking studies. And of course, you could load multiple vendor catalogs and end up with tens of thousands of fragments that match your specific criteria. Mona is easily capable of handling 100,000 compounds. So now I've demoed Mona. Just to let you know again, it's available from our website, and quickly move on. So now you've got your commercially available analogues that you might be interested in purchasing. You want to, of course, if you've got the choice of 100,000, you probably aren't going to buy them all. So now you want to have a look at evaluating those, and we'll do that by docking and scoring those fragments with FlexX. Then we're going to rescore them with the hide scoring function, which gives us an estimate of the binding affinity. And then we can interactively evaluate those ideas within our latest software package, which is called CSAR. And then I'll move on to describe how you can perhaps make this process work a lot more smoothly and with less interaction with a KNIME workflow. So I shall ask you quickly how many of you are familiar with receptor preparation in LeadIT. Because if you are mostly familiar, then I'll perhaps skip this over. So it's sort of, most of you aren't. Okay, so okay then, I'll quickly demo how easy. It maybe takes an extra 5 minutes, but it's quite easy to do. So I'll close that poll, and I'll open LeadIT. So LeadIT is a somewhat older package. It's been around for a few years now, but it's really nice for easily preparing project files for docking. So I'll just demo how easy that is. So if we go to the molecules menu, we say, okay, we want to prepare a receptor. and then we can choose a file or a PDB. I'm going to load from file because I've downloaded already. I'm going to use the same one, 106L that I showed you earlier. And then the text view of the PDB file, it just tells you a little bit of detail about it. As I mentioned, this is an activated protein kinase B with a GSK3 beta peptide and an ATP analog. Click on the next arrow, prompts me to select the chain that I want to use. I'll pick the chain in blue, that's the main kinase domain. Next, and then it's automatically selected the ligand because there is only one. Should I wish to, I could load another from file, but here I shall just accept all the defaults. Quickly jump ahead. The software has automatically protonated everything as best it estimates and decided which water molecules are important to retain within my binding site. You can change that with the waters submenu if you want to keep specific waters or not worry about specific waters. Usually the software does a really good job of assigning these automatically. So I'll just accept the defaults and move forward. And with one more click we are done. That really is a project file ready for docking. What I will do in this instance is, however, restrain my solutions to match a kinase like pharmacophore, and I'll quickly show you how to do that. So you go to the docking menu, then you pick pharmacophore and define. This gives me a choice of what kind of features I wish to use. So I'm just using the hinge binding donor acceptor motif. I'll choose the donors and pick the correct one by clicking it, and then switch over to acceptors. Again, click it, and then all the acceptors are highlighted. And here I just have to pick the correct one. Be a bit careful there. So here I want to change the default behavior because by default these are listed as being essential. You'll be able to see that in the drop-down. I'll switch those over to being optional because quite often, high-quality kinase inhibitors do only meet one of those pharmacophore points, such as the anilinoconazolines, isoquinolines only match one of those. So I'll just click on OK and accept that as the definition of my pharmacophore. So now to get the docking running, it's really simple. I just go to the docking menu, again choose Define FlexX Docking, and here I have a choice. I have to choose my library. So again, that's what I just exported from Mona. But here there's a slight problem because Mona exports those in 2D and I need 3D files for docking. So you'll have to use some tool to convert your 2D export into 3D. And we have a tool to do this. It generally only runs within 9. But if you want to generate 3D from 2D, you can do it quite easily using Marvin Space from Chemaxon. You just import and export. So select that set, and that as now click on OK. So I've got a question from, sorry, okay, I'll come back to that in a minute if I can. So now I choose my pharmacophore, the one I just defined, and I want to keep 5 poses, so I'll select from the user drop-down. And here I want to choose my base placement strategy. So there's a special single interaction scan mode for docking and scoring fragments. And essentially that is the process complete. Now all I have to do now is click on apply and dock, and that will initiate the docking procedure. So of course that's going to take a little time. We've got 197 ligands, and docking them is about 15 seconds each. So this could take a while. So what I've done is pre-prepared the output of that exercise. So I'll just switch over to that. So after maybe 20 minutes, what you get up is this. Quickly answer the question there. So here in LeadIt we have now showing all the generated docking poses, and I can flick through them and examine them should I wish. But what I really want to do is to examine those and rescore them. So I want to rescore all 713 poses. And that's really achieved with CSAR and the height scoring algorithm. So I have to first off export all my docking solutions. So I select them all, click one, and then hit Control A, and that selects all my docking solutions. And then if I right click, I can choose to export. So I'll just do that now. I'll export, save those as all docking poses. So that's all docking poses against PKB, and I'll overwrite. Okay. So docking is now complete, and as I say, for about 200 molecules, should take you 20 minutes, half an hour. So to maybe take a little step back, docking scoring is really only two questions. What we've done now is we've generated a lot of poses of a ligand in a pocket, but now we really want to understand how happy those ligands are in inverted commas, and this is the issue of scoring, and scoring is one of the hardest problems in the field, and predicting binding affinities is still an unsolved problem in terms of the general accuracy and predictability, but our HIDE algorithm does a really good job of making an estimated binding affinity range. So I've already showed you FlexX. It's one of the most accurate, most used and most cited docking programs, and as I mentioned, it has a specific mode for fragments, which I used, the single interaction scan. The little diagram there on the right shows a more classical triangle pattern matching, and the single interaction scan is useful for fragments when you have few strong interactions on a docking using the triangle match would fail. So now I want to do the last part of this slide, which is scoring using the HIDE algorithm as implemented in CSAR. And the reason I'm not doing this in LeadIt is that the HIDE scoring algorithm implemented in CSAR is an updated version and is more accurate and faster and generally much improved. So I've already shown you the demo of docking with LeadIt. So now it's just a quick question of doing rescoring with CSAR. So if I open CSAR for you, what I'll do first is just take a few moments to outline some of the features of CSAR in general. So I'll type in the code. What you see here is the opening window of CSAR is a very nice clean interface. I can download directly from the PDB server or select a file. I click on download, and what happens behind the scenes is that if the software identifies a small molecule ligand within the PDB file, that small molecule will automatically have a predicted binding affinity estimated. And here you can see our ATP analog is predicted to be active in the picomolar region. What we also have here is a yellow sphere, which is a torsional strain indicator. CSAR doesn't just estimate binding affinities, it also gives you feedback on how well the rotatable bonds in your ligand match to statistically observed patterns in the Cambridge crystallographic dataset. So here it's flagged yellow, and what we'll do now is just look at this ligand in a bit more detail. I'll do that just by clicking on it. And what happens is the view resets, and what we have now is a representation of the contribution of individual atoms to the binding potency, where a green sphere indicates that an atom is making a positive contribution to the binding affinity, and a red sphere indicates a deleterious effect for the binding affinity, and the size of that sphere represents the magnitude of that effect. If I go to the bottom right corner, we have a number of utilities where I can show atom labels, and that means you can drill down on individual atomic contributions to binding affinity. So clicking on this nitrogen atom that's involved in binding to the hinge region, you can see that when the ligand is removed from bulk solvent and binds into the active site, there's a desolvation penalty of estimated at 7.1 kilojoules per mole, but that is more than compensated for by a positive interaction with the protein at minus 8.4 kilojoules per mole. Whereas the receptor, on the other hand, went from a solvated state, and there's obviously a desolvation penalty for the receptor, but again, the receptor is gaining back a lot of potency because it's forming a nice new interaction with the ligand. I can also measure distances and delete a ligand and take a picture. But what I'd like to move on to next is just show you the torsion patterns that I talked about. So I do that just by clicking on the bottom corner. And now the spheres disappear, and what we have instead is the bonds, rotatable bonds, coloured by their match to observed patterns, statistical patterns. So here we've got lots of green bonds, a couple of yellow ones. So generally, you would say this molecule looks like its conformation is really very acceptable. What you might also see is that the dashed hydrogen bonds are different colours, and so just to explain that, a dark green hydrogen bond is a strong hydrogen bond with a optimal geometry, whereas a light green is less optimal, and the white dots suggest that there is a hydrogen bond, but it's not quite got either the correct geometry or the correct distance. For each ligand, we can start exploring some ideas. So of course this isn't really a drug-like ligand, but for the purpose of illustration, what I'll show is that if we click on the little pencil icon, which is underneath the ligand name in the panel, I can edit this ligand, and of course this oxygen atom in the middle, which I just clicked and turned pink, was making a very large negative contribution to binding affinity. And so I'll turn that atom into a sulfur, which is of course more lipophilic, and hit exit. And what happens then is that whole process is then repeated. A binding affinity estimate is calculated, and if I hit the escape key, we can now see that that oxygen atom has turned from red into a large green sphere for sulfur. So you can probably imagine if you have a crystal structure of a small molecule ligand and you want to go about modifying that ligand, you can quite readily identify atoms that are considered deleterious for binding affinity, and atoms which are making no contribution, which have no sphere at all, and atoms which are making good contributions also. So with that, I will move back to the docking of the ligands. So if here I go up to the Add Ligands menu, all I have to do is load in all docking poses PKB. And what happens then is those 700 odd ligands are loaded into CSAR, and they're all scored, and you can probably see the speed with which that scoring occurs. It's gone through the first 40 or so in less than 5 seconds. And one of the huge advantages of the height scoring algorithm is not only its performance in terms of accuracy of estimates, but also with speed. So this takes a little while, so I've pre-prepared an alternative version, which is here, where that process has been completed. So what I want to do now is, if I'm interested in picking the predicted most potent molecules, I want to sort these by potency. That's easily done. Just click on the estimated binding affinity at the top of the panel, and they're instantly resorted. So job done. And now when I created these poses, I of course generated 5 poses per molecule, and so those are duplicated within the table. If I want to collapse the entire table down to only 1 pose per molecule, I can use the toggle where I group these by molecule, and just click on that button there, and what you see is those molecules are shrunk down to only retain the best scoring pose for each one. So if I click on the top one, we can just take a quick peek. Just to explain that there's a purple sphere that appears when I rotate. That's just the center of rotation, and what I can now do is just go through these ligands and decide which ones I like and which ones I want to keep. And I can decide to keep them. There's a little favorite icon in the ligands from file panel. If I like it, I can just click it and that will turn the ligand favorite star black, and I can then quickly look through these and say, okay, I like the look of this aminopyrazole, and here you can see this aminopyrazole is predicted to make an H bond with the side chain threonine, and that side chain threonine is a valine in protein kinase A, and so I can imagine, okay, I'm going to have a good chance of identifying something a little more selective with this ligand, should I add it into my screening collection. So I'll select those, and you can just interactively go through these. Here's another pyrazole, this time with a phenyl ring, and I'll colour that by high score. You can see how most of the atoms here are green. As you'd expect, ligand efficiency, or estimated, based on the estimated binding affinity, means that this has got a big thumbs up. And if I wanted to do this exercise, I would probably select, the 20, 50 ligands that look most promising, and then export those. And the way I export those is going down to the Save Ligands to SD file icon. I'll click on that, and a little box opens, and I want to save those, just the starred ligands. So I just click on Save All Ligand marked as Favorites to File, and I can then save those, all the starred docking poses which have been rescored. I say, yes, I want to do that. So now what I have is an SD file, which I can then upload to places such as the eMolecules website. I'll just quickly show you how. And so here we just go to useful sites, eMolecules. So I could just upload a file directly into the eMolecules website of that SD file I created, and it will tell me which of those molecules are available from which vendors, and really make the ordering process really simple. So I'll just alongside this, of course, I might want to dock and score against protein kinase A, and say, or ROC2 or JSK3 beta. But it would be a lot easier if we could automate that process, because of course were I to create another project file and go through all the steps I've just gone through manually, it would be very difficult to compare predicted binding affinities and scores in this way. So it would be really nice if we could take care of the database filtering, generation of coordinates, and the docking of scoring against target 1, in this case protein kinase B, and then dock and score all those ideas against protein kinase A, and then generate an estimated binding affinity for each, and then choose either the most potent or the most selective or the most efficient, whatever I was interested in, and be able to sort them on those criteria, and then send out an order to purchase those fragments directly, and totally streamline the whole process. So in terms of taking it to the next level, what I'll demo now is how you can achieve this with a software platform called KNIME. Just be interested to find out with a little poll about how familiar or not you are all with KNIME. If you could take a moment to so we've got quite a few KNIME experts. Some have used it sometimes, and Quite some of you have never even heard of it, so quite a spread, yes. Anyway, the beautiful thing about NIME from a drug discovery perspective, as someone involved in cheminformatics, is that it is extremely well supported by a huge range of specific nodes. And within NIME, a node is just a unit of work which does a specific transformation on a data set. So we've got all kinds of useful stuff from our organization. RDKit, there's also nodes from the likes of the European Molecular Biology Laboratory, from Chemaxon, and there are many commercial nodes from most of the players in the drug discovery field, of course including BioSolveIT. So I'll close that poll, and thanks for your feedback. So at the bottom of this slide here is just a really quick workflow demo showing you load some compounds from an SD file. You generate some 3D coordinates, which you can do for free using the BioSolveIT node. Then you can dock, and then you want to score. That's of course a very simple workflow. But now, if you start thinking about what we would have to construct to achieve a fragment screen which wanted to identify selective fragments, then this is what you'd want to do. You'd want to start with a huge collection of molecules, which is e-molecules. The benefit of using e-molecules is that they make the purchasing procedure so much easier once you've decided what you want to buy. We then filter by the smarts and rule of 3 as before, generate 3D coordinates, and then prepare our receptors, do the dockings against both Protein Kinase A and Protein Kinase B, and then estimate the binding affinities, and then do the maths of comparing those binding affinities, and compare and inspect. So what you end up with, if you want to do something like that, is a workflow that looks like this, where each of those pieces is now incorporated. This is reasonably complex workflow, and I'll quickly show you what you can achieve with such workflows in quite a short time. My workflow here, this is the wrong one, use the right one, starts with loading the e-molecules collection, which is, if I mouse over here, I get 7.129 million compounds, and I then do some smarts filtering for similar kinds of kinase hinge binding motifs, and I end up with, after doing the rule of 3, about 60,000 fragments that fit my 2 criteria. I can again filter those if I want to use a tighter molecular weight limit than the rule of 3. I can specify that in the integer input node. And essentially what it does is just takes the project files that I might have prepared within LeadIt, loads those in, and then docks and scores against both Protein Kinase A and Protein Kinase B. But in this case, all it scores is a small subset of those compounds. To speed things up, I have divided my set into quarters and only kept 1 quarter. So hopefully you can see here I've picked a random 25% of fragments, and I'm going to dock and score those. And then what I can do is then use the output of that 25%, only keeping the best, look for similar molecules, and then dock and score those. So to demo what comes out the other end, I'll take the output from just docking and scoring the first 12,000 fragments. I just execute this node, and what will open up will be a new CSAR window where molecules which are predicted to be most potent against Protein Kinase B are rank ordered by predicting binding affinity. And you can see at the top of the list is this propargyl pyridine, another analyl pyridine, dimethoxy-substituted pyridine, and this one is interesting to me because hit number 5, because isoquinolines are known inhibitors of protein kinase B, so it's obviously picking out things that are known to inhibit protein kinase B, validating the approach here, that we're hunting down things that are reasonably potent and selective. And you can see again, the hydroxy group of this ligand is predicted to interact with that same threonine residue. What's also available is I can import from the calculation that's going on behind the scenes in that 9 workflow is a calculated selectivity index. So what it's done is calculate the binding affinity for pKb and compared that to pKa, and it's suggesting that this ligand here is massively selective for protein kinase B. It's very encouraging. I don't know if it's likely to end up being quite so selective, but that's what the docking and scoring suggests. And if I want to just improve the probability of hitting the right kind of fragments into my collection, then what I can do again is just select the fragments I like, maybe pick the ones that appeal to me or which make that specific interaction. I can always flick through them one by one, looking for ones which interact with that threonine residue, and hopefully here's one here that have nice torsion scores, nice predicted binding affinities. Add those to my collection, and again export those ones I've selected to SD file and upload those to wherever I wish to purchase them from. So I'll shut that one down. So the second part of this is, of course, to take a look at the end output of the workflow, which can be the most potent or the most selective, or in this one I've got the most efficient. So I'll take a look at the most potent molecules, just connect the respective node. I'll execute that node again, and that will just show me the best results, not only of the first round of screening, but also when I've gone back, taken the most potent predicted, and found similars and docked and scored those. So you can see the top scoring hit is again an allyl substituted amino pyridine, but starting to sneak into the list are things like this, which weren't so highly rated before. And this is a nice one because the 7-ASA indole framework was actually the basis of the lead series, which turned into that AstraZeneca clinical compound that I mentioned earlier. So a fragment hit like this was indeed identified in the Protein Kinase B project and turned out to be pretty good, and eventually made it all the way through to clinical testing. So just taking a quick look through these, see if I got some other. I think number 40 was interesting. Is that, so here, compounds like this, also quite interesting. So yeah, as I mentioned, you can also look at the most selective. So if my principal criteria is, okay, I have to be super selective, then we can have a look at those data. And again, you see it's super quick. Now our top scoring is pretty similar, but it's got a methyl group instead of, I think the previous one had a fluoro group. And I have to wait a moment for that to finish scoring 100. So that's now done. And again, we can open the selectivity index. And you see this is predicted to be massively selective for protein kinase B. Sometimes, if you have a docking mode for protein kinase A, what you can do is look, why is that selective? So I can connect that to protein kinase A also. So I want to, that's protein target 2, and I'll have a look at that. So what you can see here is a predicted binding mode for that azo-indole fragment. It's predicted to bind in a relatively unusual orientation. So essentially the carbonyl of the aldehyde is pointing directly down to the hinge. So perhaps it's not much surprise. This fragment obviously doesn't quite fit into the bottom of the active site. Of course, the caveat here is that we're using static protein. Any flexibility in the protein might start allowing fragments like this to sneak in and bind with more potency. But if what you're interested in doing is just stacking the deck in your favour, then I think these kinds of fragments would be a really good place to start. So what's this one here? Let's have a look. Yeah, so hopefully you can see how such workflows could really help you with enhancing your screening collection. And just to maybe talk about how long it takes to run this workflow. I've got a reasonably powerful desktop PC, and this takes the best part of 8 hours to run all the way through. So it's definitely manageable in a appropriate time frame. It doesn't require much in the way of special technology, and quite achievable in a good time. So, of course, all that is predicated on the basis that I can filter my fragment set down to a manageable number of fragments. If I have an active site which I can't use a pharmacophore constraint for, or if I have no smarts patterns that I can match in terms of pulling out similar chemical structures, then I might have to dock tens of thousands, hundreds of thousands of fragments, and that's when you'd really want to speed things up. For those kinds of situations, you'd probably require a much more powerful CPU, some kind of server-grade CPU, or cluster execution, or simply use fewer poses of each molecule per fragment. What I'll move on to for a few minutes, running relatively short on time, is how you can use similar workflows in NIME to expand your hit space. So imagine for a moment that this azo-benzothioazole is a hit against PKB. What we can do is use a similarity searching algorithm called feature trees, which we sell, to hunt down similar structures. And of course, the idea is that the similar chemical structures have a much higher probability of being active against your target. And feature trees is especially strong at making those scaffold hops. So if you want to go from something that's known to something a little bit different and perhaps patentable, feature trees is a good way to go. What you can also do is what's called a fingerprint similarity using a Tversky index. The Tversky index is very useful for pulling out superstructures of fragments. You can see in this example we've got a ethyl substituted cyanopyridine, and a lot of the top scoring outputs are also ethyl substituted cyanopyridines, because that's the focus of that algorithm, is generating superstructures. And this is of course very useful if you have a fragment hit and you want to search through zinc or e-molecules to find molecules that have that precise fragment. That you would then have a good chance you could purchase those, dock those, score those, and buy what's predicted to be nice and active. As an example of that, this is a real hit from the project, the PKB project, which is the azo-indole fragment I mentioned, where the Tversky index search pulls up as a top scoring hit the molecule itself. That's a perfect match which scores 1, and the next hit along, the pyridyl substituted azotindole retains the entire carbon and nitrogen framework of the original hit, so also scores very highly. Some of the other ones start departing a little, but you can imagine, purchasing and testing all those analogues before you even get your feet wet in the lab. You've already bought some ideas in from some vendors and can perhaps expand on your hit space. So another way you might think about expanding on your hit space is taking fragments that you've identified and looking at close analogues. This is, of course, the kind of things that medicinal chemists do all day, every day. But in terms of prioritizing those ideas, it would be nice to have some kind of system where not only would you be able to have an assessment of the predicted binding affinity, so you can avoid making perhaps molecules predicted to be less active, but also come up with some out-of-the-box thinking and ideas that might have escaped you or slipped your mind. So what we have available is something called the MedChem Wizard, which is another 9 workflow, which does essentially this. It takes a structure, in the example here is Viagra, and does a lot of the common things that medicinal chemists do in their work every day or think about doing. Things like making aromatic ring analogues, replacing rings with isosteres, diamine ring morphing, etc. So in this project we had fragments that looked a lot like this, and so what ideas does the workflow generate? I'll quickly show you. Again, this is a fairly easy to use workflow. It's designed to be straightforward. Although it looks quite complex, all the user has to do really is draw in a structure and create a project file. It does require some setting up, but what I've done, I'll just show you. If I double click on this first node, this Marvin sketch node, step 1, you can see here I've just drawn in that very same hit molecule and then run through the workflow. It generates about 600 new ideas for ligands. These have all been filtered by common kind of medchem substructure filters, Payne's filter if that's something you want to avoid, and generates, as I said, about 600 structures. Those are all then docked and scored against our target and ranked by predicted binding affinity. I'll just execute that node. What you could of course do is feed the output from this directly into the selectivity screening protocol, and instead of using fragments, just start analyzing your ideas in terms of selectivity. I haven't yet put the two workflows together, but that would of course be totally possible. So here I think we've got 250 ideas loading and rescoring. I'll just expand this window. And what you're generating is very close analogues sometimes, but amongst all these ideas are some gems. I'll just click on the first one actually, so you can see how this totally automated process has suggested that we can fit an aromatic ring in the active site from the 3 position of the indole. Just zoom in a bit so you can see that, and that specific pyridine is predicted to make a new H bond with a backbone NH3 plus of a lysine. So quite a nice idea. Other high-ranking hits are things that we did in this project. Indeed, the amine morphing kind of ideas, where you have also commercially available diamines. So imagine if I wanted to make this analog. It's got quite a nice spirocyclic structure. It's predicted to score really well, and the torsion flag is totally green. What the workflow also allows you, if I go down to this reference here, it will tell you the either the CAS number, a ChemSpider ID, or a literature reference for that specific building block for the amine morphing. So it's quite a nice way you can fast track your compound purchasing, which building blocks you want to buy, just by a quick reference to the extra column in the table. Maybe I'll have a quick hunt for some interesting ideas, and maybe go down to the... You can sort these as well by the class of the transformation. So here you've got all the amine morphing compounds listed in order. If I scroll down, look for some aromatic ring analogues perhaps, and so here we have down here some aromatic ring analogues. Here's the simple aza-indole analog, predicted to be reasonably potent again. Here's another interesting one, I think, which is the aza substituted compound, which is predicted to make a water-mediated hydrogen bond. And I don't think you would have predicted that as a first round of synthesis, but to my eye looks like quite an interesting idea. So with that, I shall halt the demo of that. So just to let you know, you can download that 9 workflow for free from our website. To do the docking and scoring, you will of course have to have a valid license for the docking and scoring components, but as part of your participation today, you do of course get a temporary license so you can evaluate. This should work in Windows 8 and OS X if you're a Mac user. And alongside that, the fragment-based design workflow I showed you today is not quite yet ready for release. If you are wanting to give it a test, I'll be happy to share that with you, but it might well not quite work perfectly, and if you were to break it, it would be nice to know what you broke and how we could fix it. On the other hand, if this all sounds a bit too complex, my role with BioSolveIT is really running these kinds of services for people that don't have the infrastructure or the know-how, and if you want some custom expert support for your discovery projects, I'll be very happy to hear for you. We do all kinds of work for people, from high throughput virtual screening all the way through to much more focused scaffold hopping studies and project support in terms of analog design, SAR analysis, and those kinds of traditional comp chem activities. So with that, I shall finish off by letting you know that you can keep in touch with what's happening in BioSolveIT world through LinkedIn or Facebook if there's software updates. As I said, you can expect an email trial license very soon. And as I said, if you're interested in running a pilot study, if you've got some project that you'd like us to work on for you, then just send me an email. I'll be happy to let you know how we think we can help and convince you to work with us. And well, there's a bit of background. So, if anyone has any questions, I'll be happy to answer them. Maybe put your hand up or just type it in the box. And if not, I will hopefully talk to you soon. So it doesn't seem as if we have... oh, we do have a question. Yes, the software is all sold by BioSolveIT, except for the very small mention that I made of the Marvin suite for generating structures. Marvin is sold is a free download from Chemaxon, but everything else, docking, scoring, similarity searching, lead IT, generating your project files, pharmacophore guided docking is all sold by BioSolveIT. I think many of the attendees today are academics. We do very attractive academic pricing, and if you're interested in licensing, following your test with the trial license, then I'm sure we'll be happy to hear from you and set you down the road. So what is the filter file format text? I don't understand exactly what you mean, Gennady. Can you help me out with a bit more specifics, or filter file? Okay. Oh, I know what you mean now. You're talking about the in Mona, aren't you? You can create the filter files within Mona directly. I'll just show you that. It's really easy. If I bring Mona up from here, and where's it hiding? It's over here. Oh, damn it. So here. So here's Mona. Within the filters window, I'll delete all my filters. I want to make a new one. So let's say I want to filter based on functional group. Let's say I want to filter all alcohols from my set and include those, but I want all alcohols that have a molecular weight between 0 and 500. So I can say, okay, create a filtered set. And then, okay, we've got 28 hits here. If I then click on the save current filter chain as a new preset with the right arrow, I can add that preset, and then if I click on that preset, I can then export that by going up to the filter presets menu and export filter presets. So I'll just do that directly. Go to webinar, XML filters, and save it in here as Gennady. So I'll show you that if I hit escape. And so that's now just sitting in this webinar folder, XML filters, and it's just a simple XML file, which you can see here. So that's how you create your own presets. You just create your custom filters, add them to the list, and then export them. What I didn't do there, I think, is didn't do that quite right. Let's just do that again. First off, you'd have to delete all these ones. You can see in the file that I created, it combined all those. So when you save your XML file, you have to remove all of those. So hopefully that. So another question is, can the error bar and the binding affinities estimates be customized by a ligand structure? Yeah, I can show you how you might do that. Let's take this version of CSAR. So within CSAR, you have the option to calibrate your predicted scores with actual scores using the button at the bottom here. Use ligands for hide performance check. So I think what you mean is, okay, I want to check the performance of hide. I want to import experimental values. I don't have experimental values for these, but I can import SDF, I don't know, let's call it one of these, and then define what units my SD activity is in, and then the software will tell you how well the software is performing in terms of the accuracy of the predicting binding estimates. Alongside that, another question from Gennady is, can you add the fog? Yes, of course you can add the fog. The fog you add by going to the edit the selected ligand and that then activates the fog. And what the fog shows you is where there's space to add a methyl group. So here, let's add a methyl. See, if it escape, you see, and you can guide your optimization that way, building into the white fog. Then we've got we could import from known ligands with known activities and assess the accuracy of things that way. Absolutely, yeah, that'd be a good way of, if you want to pre-validate whichever PDB file you've used, then you could do that, of course. Yeah. So if there are no other questions for anyone, I'll finish there. I'm happy to answer queries offline also. You have my email. We have a really great team of technical experts, and if there's anything you need to get up and running, just send me an email or post a support request via the website. So I'll just end there by thanking you for your time, and let you know that your evaluation license will be emailed to you shortly, along with a link to the video which I